So you got Imagine 50, Bev-Atezo, okay? And then you got these olaparib with durva and olaparib with pembro, okay? And then you have First, which is niraparib dostarlimab, and then Athena, which, well, we're just going to do it in maintenance. So that's 5 trials, frontline, and now I'm here to tell you there's a 6th and a 7th, and that's just with PARP. And PARP can either be done alone, all comers, called PRIMA, niraparib. So your study, Solo 1, was great, but it was only BRCA patients.
Correct.
And we want to do more than BRCA. Tell us about PRIMA now.
So PRIMA is very similar to Solo 1. It took patients who, actually a little similar but different. It took a different population of patients. As I mentioned earlier, Solo 1 were great prognosis patients. PRIMA actually took high-risk sorts of frontline patients, lots of disease, residual disease post-side reduction, so a more high risk of the high-risk advanced stage, who did have complete or partial responses to their frontline chemo, and then randomized them to maintenance niraparib or placebo. And the other difference is that it's stratified by HRD status, so not by BRCA, so HRD, so you get, of course, the BRCA patients will be stratified appropriately, but also you're stratifying based on at least the myriad assay for loss of homologous recombination.
And we want that. We like Solo 1, but it's just BRCA 25%. If we could do BRCA-like, as you said, HRD, if you will, then maybe it'd be at least half of the patients.
Right.
And who knows, we've never even studied all comers, because in the second line, it's all comers.
Right.
So,
right.
And then finally, adding PARP to chemotherapy, does that make any sense? 3005, veliparib?
We're waiting to see the results. The 3005 with veliparib, it would be the 4th PARP inhibitor. The study's been closed, and we'll have to see. We're not sure.
So it's rapidly evolving, so fast it makes your head spin. Back to our NCCN guideline expert, the #1 recommended NCCN treatment is a clinical trial.
Correct.
So you were talking to me about that prior. I think all of us are committed to clinical trials. Clinical trials are the best opportunity for discovery. I'm here to tell you that virtually every clinical trial that we've ever done in ovarian cancer, the experimental arm has never been worse. It's not always better, but virtually it's never worse. There are some isolated exceptions. You said avelumab versus PLD. Platinum-resistant avelumab wasn't quite as good as PLD, but I would argue both of them were bad. So when you talk about adding these targeted therapies, it really appears that that's the best opportunity for your patient. So I want to close with the 4th targeted therapy. We talked about anti-angiogenic, PARP inhibitors, and immuno-oncology and combinations. The next is antibody-drug conjugates. Antibody-drug conjugates, TDM1 in breast cancer, HER2/neu, and a cytotoxic payload. We have been so excited about this mirvetuximab soravtansine, which is an antibody-drug conjugate to the folate receptor, called Forward-1. We happen to have the principal investigator, and I know you know a lot about that study. It's not in the public domain, but there is some stuff in the public domain. Tell me about the design of Forward-1 and what's in the public domain.
Forward-1 is a randomized study for patients with recurrent disease, 1 to 3 prior lines of therapy, and tumor that either is medium or high to expressors of folate receptor alpha, as defined by immunohistochemistry and by a standardized test. And it randomized patients to either physician's choice chemotherapy, which was topotecan, paclitaxel, or pegylated liposomal or single agent mirvetuximab with a progression-free survival endpoint.
Antibody-drug conjugate versus physician's choice chemotherapy. I love it. And in the phase 1, the response rate was
47%.
47%. Let's get it done.
And unfortunately, the primary endpoint was negative.
Dang it.
Dang it.
Yeah. So stay tuned. We're working on it. And again, it's a superiority endpoint. So we have a number, just because it doesn't beat something doesn't mean that it's not active, and I think that's the lesson. I also am here to tell you that many of our studies are randomized 2 drugs versus 1, okay, or 3 drugs versus 2, or in the PARP inhibitor space versus observation. So it's very difficult to do one-to-one.
Right.
Okay?